Hi, and welcome to our group's video on the cell cycle, where we start at the G1 phase, go through the stages of interphase, then the stages of mitosis, and finally end with cytokinesis.
The G1 or gap1 phase is the first phase of the cell cycle. In this phase, the cell grows and prepares for division by synthesizing various proteins, such as its histones, and replicating its centrosomes. The duration of this phase can vary dramatically between different organisms, and the deregulation of this phase is very closely associated with the development of cancers. This phase contains two checkpoints, the restriction point and the G1S checkpoint. The G1 phase is actually composed of three subphases, the first two of which are mediated by external growth factors. At the end of these two phases, you come to the restriction point, which is very important to the cell cycle, as it determines whether the cell will return to G0 or proceed irreversibly towards cell division in the final stage of the G1 phase. When the cell reaches the G1S checkpoint, it confirms that the cell is ready for division. If so, then the activity of the APC complex decreases dramatically, allowing the G1 cyclin CDK complex to activate this S-CDK complex, which then pushes the cell cycle into its next phase.
Next is the S phase. And this phase, of course, comes after G1. And to move from G1 to the S phase, the cell has to pass through a restriction point where it checks that everything is on track to keep going with cell division. And two important things happen in this phase. The first is that the cell replicates its chromosomes, and the second is that it duplicates a structure called the centrosome, and the S phase is followed by G2. So the most important thing is that the cell duplicates its DNA, and I won't discuss in detail how that works. I think you can see it in other videos, but the cell replicates its chromosomes, and of course this is extremely important because both daughter cells need DNA in order to survive. They're not going to last very long if they don't have DNA. And so, of course, if you replicate each chromosome, then you're going to create pairs of them, and here they're called sister chromatids. And as you can see, the sister chromatids are linked at a point called the centromere. And later on in cell division, the centromere will be the site of another structure called the kinetochore. The other important thing that happens is that the centrosomes duplicate. The centrosomes are structures of microtubules that are used later on in the M phase. As you can see, the centrosomes are made up of a structure called the centriole, surrounded by microtubules. And of course, since it's duplicated, there's two of them.
The G2 or gap2 phase is the phase of the cell cycle that follows the S phase and precedes mitosis. In this phase, the cell makes various preparations for mitosis, including DNA repair, and has a total of 92 chromatids. This phase is not technically necessary and is skipped in some specific cells, such as cancers. It also contains the G2M checkpoint, which is the last checkpoint before mitosis. The G2M checkpoint has a very interesting pathway. The goal of this pathway is to achieve a certain threshold of active B1-CDK. The inactive B1-CDK is already present, but is inhibited by Wee1 kinase. Interestingly, once some portion of the B1-CDK has been activated, the active B1-CDK both inhibits its own inhibitor and also promotes its own promoter, which leads to a double positive feedback loop. This then results in a very rapid activation and a transition of the cell into mitosis.
Prophase starts as the nuclear membrane gradually loses its form, and the chromatin fibers start to become packaged and condensed, eventually enough to be distinguished as individual chromosomes. Each chromosome consists of two identical halves called chromatids that are connected and held together by a constriction called a centromere. Meanwhile, the condensation of material into individual units is becoming obvious at this point. The nucleolus also begins to vanish. In animal cells, a double pair of short, rod-like structures called centrioles appear, separate, and begin to move to opposite sides of the cell outside the vanishing nucleus. The cytoskeleton, which is composed of cytoplasmic microtubules, begins to disassemble, and the main component of the mitotic apparatus, the mitotic spindle, begins to form at opposite ends outside the nucleus. Late prophase or prometaphase begins with the disruption of the nuclear envelope, which is broken down into small membrane vesicles that resemble the endoplasmic reticulum and usually remain visible outside the mitotic spindle. Meanwhile, the chromosomes continue to gradually condense until they have completely formed the units that will undergo mitosis. The nucleolus also disappears during this period. The mitotic spindle microtubules are now free to enter the nuclear region, and the formation of specialized protein complexes called kinetochores begin on each centromere. These complexes become attached to some of the spindle microtubules, which are termed kinetochore microtubules. Other microtubules in the spindle, which are not attached to centromeres, are termed polar microtubules, and these help form and maintain the spindle structure along with astral microtubules, which remain outside the spindle. This signals the end of prophase.
During metaphase, chromosomes align at the cell center. Kinetochore microtubules facilitate this move to the center and align them in the metaphase plate, as we see here. Two kinetochores of each chromosome are attached to the microtubules from opposite spindle poles. This process can take additional time due to the metaphase checkpoint, which determines if all the chromosomes are attached to the spindle apparatus. Without this checkpoint, we might get more mutations which may lead to cancer. Here, the chromosomes are also visible, which allows researchers to observe if all the chromosomes are present and intact. During anaphase, the sister chromatids separate, producing daughter chromosomes. These daughter chromosomes are pulled apart to opposite poles by mitotic spindles, which are comprised of microtubules. These sister chromatids are pairs of identical copies of DNA joined at the centromere. As we know, disruptions in cell division can cause cancer. For example, lagging chromosomes in metaphase and anaphase may lead to restitution in cervical cancer. There are many variations that can occur at the chromocenters within a tumor. These abnormalities promote tumor growth.
Welcome to the next step in the process of cell division. My name is James, and I'll be reviewing the process of telophase. First, let's examine what telophase looks like so we can easily identify the main players in this process. As you can see here, the cell has started to elongate into two distinctive parts or poles. Now they'll remain connected during this phase, but are ultimately held together by what is called the cleavage furrow here in the middle. It's these microtubules in the cleavage furrow that are rearranging themselves that assist in the elongation of the cell. In each of these distinctive poles, the sister chromatids have been pulled apart, and now chromosome groups begin to reform in their respective sides. You can see these groups forming here. Now, around each of these chromosome groups, a nuclear envelope begins to form and surround the nucleus. These spindle fibers begin to dissolve into the cell's cytoplasm, and the microtubules begin to depolymerize. Once the poles have been adequately pulled apart and the respective nuclei have formed, as you can see here, the process of telophase is complete. Now, in telophase, and I guess the cell division process as a whole, really, it's notable that the two genetically identical cells have been created. Now, this is important because this is the specific type of cell is maintained, which is
vital for growth and repair. Thus, the process of cell division is inhibited. The body's ability to heal itself or even reproduce is limited.
So cytokinesis is the end of cell division. It happens in both animal and plant cells, but it happens differently. And basically, it's when the two daughter cells separate completely. In animal cells, it starts during anaphase and continues through the end of cell division, which includes telophase. And it occurs by the formation of a contractile ring made out of protein filaments. And it forms at like the equator of the two daughter cells that are separating. And it functions by pinching together the two membranes until it separates entirely. And this is shown by this image here. You could see in the middle, if you can, I apologize for the low image quality or recording quality, I don't really have control of that. The ring is in the middle and it slowly gets smaller until the two cells separate entirely. Plant cells, it happens differently though. Starting in interphase, the Golgi apparatus starts to accumulate enzymes, structural proteins, and glucose, and eventually during cell division, the Golgi apparatus will disperse into different vesicles and travel across the cell. During telophase, these vesicles are brought towards the center of the two dividing cells, and they eventually fuse together, creating something called a cell plate. The cell plate eventually becomes large enough to merge with the two cell walls on each side, obviously. And once that happens, enzymes begin to use the glucose in the cells to build up cell walls. And this happens in between the cell plate. And the cell plate actually becomes part of the membrane in the end. This image shows that. These are the vesicles gathering in the middle. Slowly they fuse together. I'll show that in a sec. And then the cell wall is created and it splits from the middle.